Good morning everyone. Welcome back to Iibs Ias Academy. Today we are going to discuss UPSC Prelims 2021 Science and Technology questions. I'm Ashok, a master's graduate from IIT Kharagpur. I have been working at Iibs Ias Academy as an assistant professor. I have been training UPSC aspirants since the past 6 years. I'm also an author. You can get my books on Amazon and Flipkart. So the first question we are going to discuss is, in nature, which of the following is or are most likely to be found surviving on the surface without soil? So, which of the following survives on a surface without soil? So, soil is not required for which of the following? The ferns, lichen, moss, mushroom. So, in this, you can go through the options. So, everyone might have seen how the mushrooms are actually grown. Everyone might have seen. So the mushrooms require the soil substratum. So the option 4 is easily eliminated. So the only ambiguity which we have is from the option B and C. Only in the option B and option C. So in this, 2 is the right answer, that is for sure. And the mosses. So this is the area where you have to focus. The mosses are the ones which survives with the soil. The moss is something which survives on the soil, whereas the lichens survive on the areas which doesn't require the soil. As you can see in the picture I have added over here, this is how the lichens look. So they grow on different surfaces and they do not require any soil. So the option B is the right answer. The next one, which of the following is used in preparing a natural mosquito repellent? So which of the following is used in preparing natural mosquito repellent? So when it comes to the mosquito repellent, so what, based on what the mosquito actually gets attracted to the body, and so to create something which the mosquito cannot find us, what is required? So in these, the options that were given is the congress grass, elephant grass, lemongrass and the nut grass. Right, these are the 4 options that were given. So lemongrass oil contains the citral at concentrations of approximately 65 to 85%. So citral is a component. Citral is something which is actually present in the citrus species, right? So the lemongrass has this kind of citral which makes the mosquito not find the body. So the mosquitoes, or if you take the human body into the perspective, the bacteria which grow on our body also contributes to attract the mosquitoes. That means the fluids which are released, the aura or the smell that is released through the body, and also the carbon dioxide which is being exhaled. So these are the things which actually attract the mosquito to actually come to the body. So when you apply this lemongrass to the body, so the mosquitoes cannot really find you. So it reduces the mosquito bite by 40%. It reduces the mosquito bite by 40%. But you cannot use the concentrated lemongrass or concentrated citrals. You cannot apply it on the body directly, so you have to dilute it to the required proportions, and then you can use. So the lemongrass is something which is a natural mosquito repellent. The next one, consider the following kinds of organism: copepods, cyanobacteria, diatoms and foraminifera. So these are the 4 things, and which of the above are the primary producers in the food chain of the oceans? The primary producers in the food chain. So when it comes to the primary producers in the food chain in the oceans, so the primary producers are something which performs photosynthesis, and they act accordingly, and they produce food through metabolizing carbon dioxide and sunlight. As you can see here, the cyanobacteria and the diatoms are the 2 organisms which are photosynthetic, cyanobacteria and diatoms. So most of the people know about the cyanobacteria, which we also call it as blue-green algae. We call it as blue-green algae, and the diatoms are also the photosynthetic, microorganisms, unicellular microorganisms. So the cyanobacteria and the diatoms. So option 2 and 3 is the right answer. So the copepods is the primary consumer, as you can see, and the diatoms are the cyanobacteria or the producers, and foraminifera are the single cell organisms which belong to the protista. The protista are unicellular organisms which where all the amoebas or the small the granular the organisms with the granular ectoplasm comes into this area, right? So these are foraminifera, single cellular eukaryotic organisms, please do remember, that is foraminifera. So the diatoms and the cyanobacteria are the primary producers in the aquatic ecosystem or the saltwater ecosystem. Next, consider the following animals: hedgehog, marmot, and pangolin. To reduce the chance of being captured by predators, which of the following above organisms roll up and protect their vulnerable parts? So, among these organisms, among these animals, which of the animals actually roll in such a way that the sensitive areas are covered so that the predators cannot eat them? So, in this, the hedgehog and the pangolin are the organisms or the animals which roll themselves so that the outer exoskeleton is surfaced so that the animal, the predator, cannot prey on them. You can see the pictures, right? You can see this, the hedgehog and the pangolin. This is the hedgehog and this is the pangolin. So they roll up. So the hedgehog has the spines as the exoskeleton, and this has the scales. So because the abdomen, the surface, the dorsal area, the ventral area is sensitive, so they roll themselves up so that the exoskeleton is exposed so that the predators cannot prey on them. Next one, which of the following is a filter feeder? Which of the following is a filter feeder? The filter feeding mechanism is something where all the impurities in the water are actually filtered and the water is carried through the body. So in the waters, obviously there will be sediments. So the filter feeding mechanism is present in few fishes and animals, the few aquatic animals, that the sediments or the soil particles were actually filtered from entering the body. So the one which has the filter feeding mechanism is the oyster, as you can see here. The oysters or the surface here at the gills, they have the filter feeding mechanism that they filter the particles from entering. So to form the pearls, what we do, we open it a bit and we add small sand particles so that the sand particles which in turn form into pearls. So the oysters have this filter feeding mechanism. In a pressure cooker, the temperature at which the food is cooked depends mainly upon which of the following. The first one, the area of the hole in the lid, the temperature of the flame, and the weight of the lid. Right? So the pressure cookers work on the principle of high pressure and increasing the boiling point of the liquid or the water. So the water usually boils at 100 degrees. So what we are doing, we are increasing the pressure so that the water will not be evaporated at that particular temperature. So for the pressure cooker, the temperature increases beyond 100 degrees. Beyond 100 degrees, irrespective of what the flame you put on, is because of area of the hole in the lid, so that the pressure can be maintained. If the area of the hole in the lid is more, the pressure will be released automatically and the temperature cannot be maintained within the pressure cooker. So the area of the hole in the lid is an important factor, and the weight of a lid is also an important factor. So that's why for the cookers, the weight of the lid matters the most. They are really strong, and if the weight of the lid is not so high, we see the spring action, so that will protect the lid from opening without having any of the exposure or any of the opening, so that the pressure can be maintained within the pressure cooker. So the option 1 and 3 is the right answer. Option 1 and 3 is the right answer. The next one, consider the following: the bacteria, fungi, and viruses. Which of the above can be cultured in the artificial synthetic medium? Artificial synthetic medium is something where we provide the carbon source, nitrogen source, oxygen source, and hydrogen source. Carbon, nitrogen, oxygen, hydrogen. So all these four sources will be given to the organism synthetically, artificially, so that the organism can grow. So which of the animals can grow in the artificial medium? So the bacteria and fungi can grow in the artificial medium, whereas the viruses need a living host. The viruses need a living host for the survival. The viruses need a living host for the survival. So if I am alive, the viruses can survive within me. If I die, the viruses die. They can only be transmitted through the living host. So that's why the option 1 and 2 only, the option A, only 1 and 2, the bacteria and fungi can be cultured in the artificial synthetic medium. Consider the following statements. Adenoviruses have single-stranded DNA genomes, whereas retroviruses have double-stranded DNA genomes. Common cold is sometimes caused by an adenovirus, whereas AIDS is caused by retrovirus. Adenoviruses have single-stranded DNA genomes, whereas the retroviruses have double-stranded DNA genomes. And common cold is sometimes caused by adenovirus, whereas AIDS is caused by retroviruses. So the retroviruses or the RNA viruses. Retroviruses or single-stranded RNA viruses. So this is one thing which we know. And adenoviruses, recently we are talking about the coronaviruses, right? Which cause flu-like symptoms. The common cold is actually caused by adenoviruses. Right? Common cold is caused by adenoviruses, whereas AIDS is caused by retroviruses. Option 2 is correct. So coming back to the first statement, adenoviruses have single-stranded DNA. That is a wrong statement. Adenoviruses have double-stranded DNA. Double-stranded DNA, whereas retroviruses have single-stranded RNA. Retroviruses have single-stranded RNA as a genetic material. So option 1 is wrong. Only option 2 is correct. Only option 2 is correct. Okay. Single-stranded RNA. This is double-stranded DNA. Bisphenol A cause of concern is structural or key component of in the manufacturing of which of the following kind of plastics? The bisphenol A is a cause of concern. This is the area of the current issues. Recently, this question has actually come. This is the toxic agent which is being released into the atmosphere, causing the pollution and different diseases. So the bisphenol A is actually released or the main component in the manufacturing of polycarbonate plastic. Polycarbonate plastic. So whatever the plastic which we are using, it is mostly the polycarbonate plastic. So in the manufacturing of polycarbonate, the bisphenol A is being released, which is a structural component of this polycarbonate plastic. Right? And the bisphenol A mimics the estrogen. The bisphenol A mimics the estrogen, which is found in the feeding bottles of the infant. The feeding bottles of the infants has the estrogen. Estrogen is also known as love hormone. In 2018, the government of India has banned the supply of estrogen without any authorization, because the estrogen has been used for different purposes, like to increase the size of the vegetables, or to for forced delivery, and also the estrogen is being used for the animals to produce more milk. So that's why the estrogen has been banned. And the bisphenol A is considered to be the carcinogenic or the cancer causing. The bisphenol A is considered as carcinogenic or as we speak the cancer causing and linked with lower IQ in the newborns. So that's why it is most dangerous, and this has been in the news recently. And triclosan, considered harmful when exposed to high levels for long time, is most likely present in which of the following? Food preservatives, food wrapping substances, reused plastic containers, and toiletries. Triclosan. So this is also has been in the news recently because with the COVID-19, there has been an increased usage of sanitizers. There has been an increased usage of sanitizers. So the triclosan is present in these hand sanitizers and the hand washes, or we call them toiletries. We call them toiletries. So this has been considered harmful when exposed to high levels for a longer period of time. So the increased use of hand sanitizers can actually be harmful to you. So please stop using it. As you can see in the news, due to COVID hand washing protocols, a study by IIT Hyderabad had found its harmful effects, right? It is, and also the excess use of hand sanitizers actually kill the good bacteria present in the body. So, which of the following is the reason why astronomical distances are measured in light years? So, the next question is which of the following is the reason why astronomical distances are measured in light years? The light year is something where the time taken by a light to travel one distance traveled by a light in 1 year is called light year. So, it is used to measure the distance traveled by light for 1 year. We call it light year. So, which of the following is the reason for taking this light as a measure to as a measure to measure for distance, right? So the first one, the distance among stellar bodies do not change. That is fake. That is wrong. So the stellar bodies, as we speak about the Big Bang theory, the spaces between the stars or spaces between the galaxies is actually increasing, right? And the gravity of the stellar bodies do not change. The gravity do change with the change in the mass, and the light always travels in the straight line. So no, it has both the particle nature and the wave nature, and the speed of the light is always same. Yes, right? As per the Einstein's theory of relativity, he has considered the speed of the light to be the same always. So that's why we are taking the same measure and we are trying to measure the distance using light. Speed of a light is always the same. Option D. Water can dissolve more substances than any other liquid because the water can dissolve more substances than any other liquid because the reason is it has both positive charge and the negative charge. As you can see here, the water is considered polar molecule because it has positive and negative, right? We have hydrogen as positive, we have oxygen negative. So that's why the water is a universal solvent. Water is regarded as a universal solvent, right? So because of its bipolar nature or dipolar nature, both positive and the negative charge. With reference to the street lighting, how do sodium lamps differ from LED lamps? So with reference to the street lighting, how do sodium lamps differ from LED lamps? So since the past many decades, we have been using sodium lamps, and recently we are replacing the sodium lamps with the LED lamps. What is the reason behind it? Right? So how are they different? The first statement, the sodium lamps produce light in 360 degrees, but it is not so in the case of LED lamps. Sodium lamps produce the light in the 360 degrees, whereas the LED lamps focus on only one side. The next statement, as street lights, sodium lamps have longer lifespan than the LED lamps. And the third statement, the spectrum of the visible light from the sodium lamps is almost monochromatic, whereas LED lamps often offer significant color advantage in street lighting. So when you consider the first statement, yes, the sodium lamps produce the light in all the directions, whereas LED lamps are focused only in one direction, so only towards the road, so that there won't be the light pollution. There won't be any light pollution because it is totally focused in the on the road. It is not 360 degrees. Next one, the street lights. As street lights, sodium lamps have longer life span than the LED lamps. That is a wrong statement. LEDs are more famous because it is cost effective. It is the manufacturing costs are very less, and also it has longer shelf life, longer lifespan. And the spectrum present, spectrum that is coming from the sodium lamps, as you might have seen in the street lamps, it is mostly yellowish color, so it is monochromatic light, whereas LED lamps, it is a white color, that means it is a polychromatic light. So the option 1 and 3 are only correct. So option 1 and 3 are only correct. The term ACE2 is talked about in the context of, the ACE2 has been in the in the news recently because the ACE2 is a marker, important marker on the viruses, and this is the area which is actually binding to the cell, right? So that ACE2 has been used as a potential diagnostic and the prognostic biomarker for chronic inflammatory lung diseases. For the lung diseases, this is regarded as an important marker. That means to identify whether the person or the people are infected with these viruses or not. So the identification of the ACE2 will actually result in the control the spread of viral diseases. So once you identify, once you go for the diagnostic or the prognosis of the diseases, so you can directly control the spread of viral diseases. So the term ACE2 is talked about in the context of spread of viral diseases. In the context of hereditary diseases, consider the following statements. The passing on mitochondrial diseases from parent to child can be prevented by mitochondrial replacement therapy either before or after in vitro fertilization of the egg. The child inherits mitochondrial diseases entirely from the mother, not from the father. So the mitochondria, which is also known as the powerhouse of the cell, has a unique feature. It has its own genetic material. So if there are any mutations in this mitochondrial genetic material, so they cause mitochondrial based diseases. So the mitochondria has to be replaced. Mitochondria has to be replaced. How do you replace it? So the mitochondria replaced either before or after the in vitro fertilization. That means you can take an egg, and remove the nucleus and put it in the healthy egg, which has healthy mitochondria, and you fuse it with the sperm, form a zygote. So in that way also you can do, or else after the fusion, after the zygote formation, you take the zygotic cell and you remove the entire nucleus of the zygote, or total 46 chromosomes you remove, and put it in the healthy egg. So in that way also you can do. So it can be done before or after the in vitro fertilization, and the child inherits the mitochondrial disease entirely from the mother, not from the father. Yes, even though the sperm contains more amount of mitochondria for its survival for 72 hours, but the mitochondria will not be transferred into the egg during the fertilization. So all the mitochondria present in our body is solely contributed by our mother. So the mitochondrial diseases are inherited only from the mother but not the father. So both the statements are true. So the next one, with reference to the recent developments regarding recombinant vector vaccines, consider the following statements. So the vaccines. So with the rise in COVID pandemic, so we have had designed the vaccines. The vaccines are something which elicit the immune response. The vaccines are something which elicit the immune response against the pathogen, so that if the pathogen enters into our body, our immune system will be ready to fight the pathogen, right? So we have different types of vaccines available. So we have the heat attenuated vaccines, inactivated vaccines, we have the subunit vaccine, we have the toxoid vaccine, we have the recombinant vaccine, we have DNA vaccines. So these are the wide array of the vaccines that are available, right? In this, we are talking about recombinant vector vaccines. In this, vector is also an important term. The vector is something which carries the vaccine. The vector is something which carries the vaccine. The Coming back to the statements, the genetic engineering is applied in the development of these vaccines. Bacteria and viruses are used as vectors, right? Yes, definitely for recombination purposes we use genetic engineering tools, right? We use genetic engineering tools for the development of these vaccines. Yes, the statement is absolutely true. And the vectors, the carriers for these kind of recombinant vaccines, yes, the bacteria and the viruses are used so that the live bacteria and the viruses can stimulate the immune response way efficiently than the normal inactivated the vaccines. So that's why yes, for as a vector, the bacteria and the viruses are used, which cannot cause the disease but elicit the immune response properly. And Bolgard 1 and the Bolgard 2 technologies are mentioned in the context of what? The Bolgard, as you can see the statements, colonial propagation, development of genetically modified to the production of plant growth substances, the production of biofertilizers, right? So the Bolgard 1 and the Bolgard 2, or the insecticidal plant varieties, they are the genetically modified crop plants, right? They are the genetically modified crop plants which act against the ball insect, which can act against the ball insect. So they are genetically modified crop plants, right? So they can act against the ball guard cotton, provides inbuilt protection for the cotton against American bollworm, right? Against the American bollworm. So these genetically modified plants are actually obtained from the Bacillus thuringiensis. Bacillus thuringiensis, that's why we call them BT cotton or the BT brinjal or the BT mustard. BT means Bacillus thuringiensis. Bacillus thuringiensis. So the Bolgard 1 and Bolgard 2 technologies are mentioned in the context of development of genetically modified crop plants. Genetically modified crop plants. So in the next class, we are going to discuss the environment and ecology. Please do like, share, subscribe, and comment. And if you have any queries, please put down in the comments below so that I can discuss in the next video. Thank you. Bye.